Hi, in this video we are going to talk about RT-PCR. You can find written notes on what we are going to discuss today at medicinecanbeeasy.com. The link is also in the description. So what are you going to learn in this video? I am going to explain what RT-PCR is and how it works. We are also going to compare standard PCR and RT-PCR and discuss the applications of the method. Okay, so let's start. RT-PCR, or reverse transcriptase polymerase chain reaction, is another modification of standard PCR, which uses an enzyme called reverse transcriptase to transcribe RNA into cDNA, the so-called complementary DNA, which is then amplified using the standard PCR method. Reverse transcriptase is mainly found in retroviruses, for example in HIV, which after infecting a host cell use the enzyme to transcribe their RNA into cDNA, which is then incorporated into the genome of the infected cell. Apart from reverse transcriptase and RNA template, RT-PCR uses the same basic components as conventional PCR: DNA polymerase, primers, dNTPs, and a buffer. To carry out an RT-PCR, first a primer must bind to the RNA molecule, which allows reverse transcriptase to synthesize a new cDNA strand. The original RNA is then degraded by another enzyme, RNase H, and a second cDNA chain is synthesized by DNA polymerase. Then the reaction continues as conventional PCR. The two strands are separated using a process called denaturation, when we simply heat the DNA to about 90 degrees Celsius, which causes the two strands to separate. Once the two strands are separated, we can proceed to the second step, which is called annealing. During this step, the temperature is lowered to about 40 to 65 degrees Celsius, and the primers attach, anneal, to the two DNA strands, more specifically to the target DNA sequence we want to amplify, we want to copy. And in the final step called extension, we again heat the mixture to about 70 degrees Celsius, and DNA polymerase synthesizes the new DNA strands in the 5' to 3' direction. This process is then repeated many times, resulting in a huge increase in the reaction product. So that was the reaction itself, and now let's take a look at how we can apply the technique. Today, perhaps the most well-known application of RT-PCR is the diagnosis of diseases caused by RNA viruses. To do this, the viral RNA is transcribed into cDNA by reverse transcriptase and amplified after complementary primers bind to their target sequence. The amplified viral cDNA then confirms the viral infection. However, there are many other applications of RT-PCR. For example, RT-PCR is used to analyze gene expression in cells. The mRNA molecules are converted into cDNA, and these can then be analyzed. So this is it. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos on genetics or medicine in general, please subscribe. Thank you again, and see you soon.